Hello students, welcome to lecture 1 for cell technology. There will be a set of 3 videos for this lecture that will cover the introduction to mammalian cell technology. Cell technology focuses specifically on mammalian cell culture, and it is very different from bacterial cell culture that perhaps you may be familiar with. So what is exactly mammalian cell culture? Mammalian cell culture actually involves growing and maintaining cells which are derived from mammals in a controlled environment outside of their natural habitat. So typically in a laboratory setting. The conditions within the culture system are normally very carefully controlled to mimic that physiological environment of the cells when they are found within the mammal's body as closely as possible. So this includes controlling factors such as temperature, humidity, pH, oxygen concentration, nutrient availability, and definitely in a very contamination-free condition kind of environment. So why exactly is mammalian cell culture important? Mammalian cell culture is actually one of the very major tools used by scientists around the world in the field of cellular and molecular biology. It is very crucial for studying the normal physiology and biochemistry of cells, studying the effects of many different kinds of drugs or toxic compounds on the cells. You can also use it to study mutagenesis and carcinogenesis. It is also used in drug screening and development, and also large-scale manufacturing of biological compounds such as vaccines. So mammalian cell culture are pretty much being used a lot in all these different kinds of settings. It allows basically scientists to investigate very complex biological phenomena in a very controlled setting, and it provides also a platform for them to develop new treatments and therapies. There are actually a lot of information that you can gather from experiments that uses mammalian cell culture. So here in this schematic figure, you can see some of the examples. One of them is genetics. You can actually study the genetic makeup of the mammalian cells in order for you to understand their structure, function, and also regulation. You can use cells to be transfected by what it means is that you introduce foreign genetic materials such as DNA or RNA molecules into these cells, and you can study a lot of things from it. That you can also do transformation where you can actually transform these mammalian cells so that they are able to proliferate indefinitely. Um, at the same time, you can also look into intracellular flux, where you actually try to understand how and also the rate at which the metabolites flow through these biochemical pathways that are taking place within a cell. Um, Basically, these intracellular flux allow you to study the movement of molecules from one metabolic intermediate to another as they undergo various different kinds of enzymatic reactions to generate energy or synthesize biomolecules of your interest, and also to even maintain the cellular functions. You can also gain certain knowledge in the cell-to-cell interaction, which is the communication and also the physical interactions that actually takes place between individual cells within a multicellular organism. So these interactions are very important because they help to coordinate cellular behaviors, they help to direct or organize tissues and organs formation, and also maintaining physiological functions. So cell-to-cell interactions basically can actually take place in many different kinds of forms, and also it involves a range of molecular mechanisms that you can study. These include like the direct physical contact between adjacent cells, or even signaling through the different soluble molecules like growth factors, cytokines, and even interactions with the extracellular matrix. That means the environment around the cells. You can also look at intracellular activity, what is happening inside, right? The various biochemical and physiological processes that are actually taking place inside the cells. These activities are very, very essential because they help to maintain the cellular homeostasis. They help the cells to perform their functions, and also they guide the cells to respond, how to respond to different environmental cues. So you can actually study this in mammalian cell cultures, right? You can look at metabolism, gene expression, signal transduction, protein synthesis, and even organelle function that are found within the cells. You can also look at the environmental interaction. Okay, so what does this mean? It is actually the interaction between the cells that you are culturing and the surrounding environment, which includes, like for example, different cell culture media that you use, the substrate that you use. You can also play around with the physical conditions and study how the cells actually respond. So these interactions are actually very crucial when you are growing cells in culture in the lab, because these interactions can play a role in maintaining the cell viability, the proliferation of the cells, the differentiation of the cells if you are growing stem cells, and also functionality of the cells in vitro, that means in culture. Okay, so these environmental factors that you are playing around within the cell culture can actually influence all these things, right? The cell behavior, the physiology, and this includes all the gene expression, the metabolism in the cells, and the different cell signaling that's happening in the cells. And also you can look at how the morphology of the cells changes over time. Lastly is you can look at cell products, meaning what are the various compounds that are secreted by the cells. Okay, cells are natural biofactories. They are able to release a wide range of molecules, okay, such as proteins, hormones, enzymes, antibodies. So all these secretions occurs when these molecules are actually synthesized within the cell, and then they are transported out of the cell membrane into that extracellular environment around them. Okay, so this is important because it can also guide you downstream how to actually design your bioreactor, where you include factors like such as agitation, aeration, pH control, and how you regulate temperature and nutrient supply to actually optimize your mammalian cell growth and productivity. So all these points that is listed in this slide are very useful knowledge that we can actually gain from a mammalian cell culture. So before we actually jump into the actual cell culture techniques, we have to, of course, first understand the details of a cell culture laboratory. So I've divided this into three subtopics. First is the setup and design of a cell culture lab. Second is the basic equipment and lab requirements. And also lastly is the basic maintenance of cell culture lab. The objectives for the three subtopics for this lecture one are to define what is a good setup and design of a cell culture lab. Second, to list the essential and beneficial equipments for setting up the basic culture lab, and also to understand and to be able to describe how some of these key equipments function. Thirdly is to describe the care and maintenance of these equipments and also the cell culture lab in general. Okay, in the setup and design of cell culture lab, a cell culture lab should always, okay, allow sterile handling of cultured cells with extremely minimal level of contamination. Sterile means very, very clean, absolutely clean, highest level of cleanliness. All right, why? Why is this important? Let's look at the cell division rates for a bacteria versus a mammalian cell. So for example, E. coli, it divides what? About 15 to 20 minutes. What about mammalian cell? They divide approximately about 24 hours in general. We're speaking in general. So let's imagine one bacterial cell accidentally fell into your cell culture flask while you're working. And this flask contains 2 million human breast cancer cells, for example. And one bacteria drop in, okay, and this bacteria divides about once every hour. So what's going to happen? So after 24 hours, what's going to happen? The number of mammalian cells is going to be doubled to 4 million. And how about the bacterial cells? Using the formula, you're going to have about 16 million or 17 million of these bacterial cells. What does that mean? You have a bacterial contamination in your cell culture flask, because the growth rate of bacterial or fungal cells is usually so much greater than that of a mammalian cells that no level of contamination can be tolerated in an animal cell culture. How does bacterial contamination look like? So here is human embryonic kidney 293 cell line, or we call it in short HEK 293. They are adherent cells. They grow on the surface of the flask. It is actually contaminated with E. coli. So you can't really see this E. coli at low magnification, right? At picture A. But if you were to look upon further magnification in picture B, you can see all these tiny specks of E. coli. So you can only view bacterial cells usually at higher magnification. So that's generally how bacterial cells contaminate your culture. So if you ever in the lab, you're growing your cells, you want to check if you have any bacterial contamination, you have to use the microscope and look up at a higher magnification and try to find all these little rod shaped like, and they're usually moving a little bit. Okay, they're moving around. Here are examples of images that showing cells contaminated by yeast and fungus. So they all actually look very different, but these are the three main contamination: bacteria, yeast, and fungal. So if we researchers are working in a mammalian cell culture laboratory, do we really have to totally gown up like the pictures shown here? Is it really necessary to design a lab to be totally free of potentially contaminating microorganisms? Is that even almost possible? What do you think? Of course, it is impractical to design a cell culture lab that is totally free of any potentially contaminating microorganisms. Almost impossible, right? Generally, the cell culture labs around the world, it is generally designed to minimize as much as possible the risk of contamination for an experienced scientist. So for all of you that are taking cell tech, what's going to happen is we're going to try our very best to train ourselves up to be as cautious as possible, to have really good techniques, right? To be like an experienced cell culture scientist. So the maintenance of these non-contaminated cell cultures that you're going to be growing, right? To maintain that your cultures are non-contaminated, it is depending on two key things. One is your good experimental techniques, and second is your ability to maintain that clean, germ-free environment where you are working with your cell cultures, so that you don't need to even gown up like this every time when we go for our practical sessions. There are several considerations that need to be taken into account when you are planning the layout of a cell culture lab. So the first one here, you can see, it is very important that you have a good physical separation of your sterile handling area and also from the areas for wash up and for sterilization. Ideally, okay, in an ideal setting, these two sections should be separated in different rooms of the laboratory. Second, your cell culture facilities or the area that you are actually doing your sterile handling, they are often located in a smaller room. Reason being to cut down that traffic of personnel walking around. Okay, and thirdly, you will have a laminar flow hood, or can also be called as the cell culture hood, your incubator, and your inverted microscope generally these three items, these are key items in cell culture lab. They should be positioned as close together. All right. Reason is you want to minimize that physical transfer of your cell culture flask from one point to the other point. So you have to have all these three equipments very close together. So here is a simple picture. You can see where the clean area here, you have the microscope, you have the carbon dioxide incubator, and your BSC, your biosafety cabinet or laminar flow hood in one section. And then you have all the wash up and the preparation areas on the other section. That's where you'll find your water purification system, your sink, your washing things, your autoclave machine. So they are separated, all right? So for example, if you have a small tissue culture or cell culture laboratory, this is generally a suggested layout, okay, for a self-contained cell culture lab that's generally used by 2 to 3 people. Okay, if you just have a single room, you usually have to create a sterility gradient, meaning the clean area for sterile handling is located at one end of the room, preferably furthest from the door where you go in and out, okay? And your wash up and sterilization facilities is on the other end of the room, okay? And then you can have in between the clean and also so-called the unclean area, you can have your storage or incubator, and also some preparation tables for you to prep things before you want to go into the cell culture hood, okay? This one is, I would call it the medium-sized cell culture lab. All right, suitable mostly for 5 to 6 people. However, you can see the clean area is separated from the washing up and the preparation facility. The washing up and preparation facility is located someplace else. So you see the clean area, you can see all the standard, the laminar flow hood, okay? You will have all the inverted microscope. You will have the key. You will see similar pattern or similar design from small, medium to even large tissue culture lab that I will show next on my next slide. So here is a picture, an example of a large cell culture lab, generally suited for 20 to 30 people. However, you can see the clean section is totally separated. They have from the washing up, sterilization, and preparation area. Those three areas are actually in another room, right, adjacent to the clean room. The next slide, I will show you an example, just an example how it looks like. So here is an example of a real lab to show you the separation of the clean sterile area, which is on your left, and the non-sterile area, which is on your right. So you can see the sink, the water bath, the prep area, it's all on the right side, which is considered the non-sterile area. Okay, so you can see there is this gradient, right? Gradient of sterility from the left, clean, all the way to the right, which is unclean. Okay, so generally the design is typically similar, okay, that kind of layout, all right? So I hope you get the understanding of how you want to set up and design your cell culture lab. If you ever have that experience in the future at your workplace where you're helping a company to design a cell culture lab, so here you go, these are some of the key pointers. With this, I wrap up the section on the 1.1, which is the setup and design of a cell culture lab. Hello students, this lecture will cover point 1.2 on the basic equipments and lab requirements. At the end of this lecture, you should be able to list the essential and beneficial equipments for setting up the basic culture lab and also describe the functions of some key equipments. There are in general 7 main functions in a cell culture lab. They are split between the sterile or the clean areas or the non-sterile or the non-clean areas. The functions in the sterile area include sterile handling, incubation, and observation. while the functions in the non-sterile areas would include preparation, wash-up, sterilization, and storage. Generally, in the sterile or the clean area, it is always a very quiet part of the lab, and it is very restricted for only cell culture work. Okay, the reason why it is always found in the quiet part of the lab because you do not want to have too much traffic that may cause dust or drafts that will be flowing around there. All right, so in the clean area, you will generally find the cell culture hood or the laminar flow hood where you will perform all your sterile handling work. And then nearby, you will see the incubator for your cellular incubation, for you to grow your cells in, and also the microscope for you to observe your cells. Cell culture hoods are the key basic equipments for your sterile handling work. What does it actually do? These culture hoods, they are able to filter sterilize the air that is passing over the work area that you are working in, so that the work environment within that culture hood is protected from dust and also bacterial or fungal contaminants. Hence, these culture hoods are very, very important, okay, because they actually provide an aseptic work area inside, right? And it also allows the containment of any infectious splashes or aerosols that could be generated by your microbiological procedures in your cells, right? And then at the same time, these hoods, they are designed to have very specific airflow patterns. Okay, these airflow patterns are important because they actually help to maintain that clean, sterile environment for your cell culture work in the hood. The airflow in these hoods are key because it serves to protect both user, that means yourself, from any infectious materials that are generated from the cell cultures that could be exposed to you. So you are protected from that. And at the same time, the cell cultures in the culture hood is also protected from any types of contamination. As I mentioned previously, the major advantage of working in a laminar flow or cell culture hood is that the working environment that you are working in is protected from dust and also contamination by a constant stable flow of filtered air which passes over the work surface. There are two types of flow hood, the horizontal and the vertical. In the horizontal flow hood, the air is drawn from above and they are then filtered via the HEPA filter and then they are forced out to the front of the working area of the hood. This air is not recirculated. However, in the vertical flow hood, it draws in the air through the front of the working area into the vent system of the cell culture hood at the front of the working surface. And then it goes up and then it is filtered. 80% of the air is recirculated. So this filtered air is recirculated and then it enters back into the working area while the remainder of the air is then expelled out. Here is a real image of a horizontal flow hood. As you can see from this image, this horizontal hood has fans behind the vents at the back of the working surface and they actually blow this air outwards, all right? So this flow hood actually provides a clean and particle-free workspace for the user and also any materials that are placed on the work surface. Here is a real image of a vertical flow hood. As you can see here, this vertical flow hood has intake vents at the front of the working surface and also at the back, at the wall of the working surface. This particular flow hood minimizes the risk of contaminants which can settle onto the work surface as gravity helps to direct the particles away from the workspace. This kind of flow hood also provides significant protection to the user and the cell culture, better than the horizontal flow hood. In the research world, there are four common types of cell culture hoods that you can find in the labs. All right, you can find the laminar flow hood, class 1 biological safety cabinet, class 2 biological safety cabinet, and also class 3 biological safety cabinet. Laminar flow hood is the basic culture hood that you can find. They do not provide personnel or environmental protection as well as the other types of cell culture hoods. They only provide a clean, sterile workspace. It can either have a vertical or a horizontal airflow. However, in cell culture labs, you'll always find the vertical airflow laminar flow hood. They are very suitable for applications that require aseptic techniques such as cell culture, tissue culture, or even microbiological manipulations. However, they are not suitable for working with hazard or infectious materials unless there are additional containment measures that are in place. Class 1 biological safety cabinet provides significant levels to personnel and environmental protection, but not exactly to the product. That means the items that you're working with. Typically, they have a vertical airflow and they are designed with an open front with inward airflow, which provides personnel protection, and HEPA filtered exhaust air, which is for the environmental protection. They are usually used for working with low to moderate risk agents or when protection is primarily needed for the operator and also the environment. Class 2 biological safety cabinet provides significant levels of protection to personnel, environment, and also the product. It usually have a vertical or horizontal laminar airflow pattern depending on the designs of the safety cabinet, and it is designed with an open front with an inward airflow which is providing personnel protection, a downward HEPA filtered laminar airflow which is for product protection, and also a HEPA filtered exhaust air which is for the environmental protection. They are very suitable for handling potentially hazardous materials such as primate derived cultures or cultures that you have virally infected them, or radioisotopes, carcinogenic cells, or even toxic reagents. Class 3 biological safety cabinet is the highest type of cabinet in terms of the strictness in the level of containment. Okay, it offers the maximum protection to personnel, environment, and product. Okay, if you see a class 3 cabinet, it is totally enclosed, right? Enclosed, and it's operated through glove ports, so you can only enter your hands inside. And they have other sealed mechanisms, okay, and they have this leak tight construction, and nothing gets leaked out of there, okay. It is sometimes they have a gas tight system and generally have a vertical airflow pattern. This cabinet is maintained under negative pressure and the supply air is drawn in through HEPA filters. So all air that gets in is through HEPA filters. They are filtered, okay? And the exhaust air is all treated with either a double HEPA filtration or a single HEPA filtration followed by air incineration, and then it is exhausted outside. Okay, so this type of biosafety cabinet is only good or being used primarily with if you are working with human pathogens and other BSL 3 or 4 kind of materials. So these are really infectious, hazardous type, okay, highest level. So you use class 3 biological safety cabinet to provide that maximum protection. So personnel protected, environment protected, and the product is also protected from any kinds of contaminants. So really highest maximum protection. The next basic equipment that you must really know is the incubators for cell cultures, generally known as the 37 degrees carbon dioxide incubator. They are important because they have to maintain that environment which is optimal for cell growth. So the three parameters that are mainly measured, controlled, and reliably maintained are the temperature, humidity, and carbon dioxide concentration. Generally, for a mammalian cell culture incubator, the incubator is set at 37 degrees Celsius and it has a relative humidity level of 95 degrees and with a 5% carbon dioxide concentration. The atmosphere within the incubator is normally humidified by means of a tray that generally you place at the bottom of the incubator, and you have a fan inside the incubator to ensure the distribution of humidity around the chamber is pretty much even. The next equipment is the one that you need for observation of your cells. Okay, so a good inverted microscope is really essential in any cell culture lab because you need to use it to allow you to visualize cultures in your flask or in your plates, and also for cell counting. There are several objectives of these microscope. They are 4x, 10x, 20x, and sometimes you get the 40x, and it should have a good phase contrast feature in order for you to be able to see your cells clearly. Okay, so this is an example of an inverted microscope. It has a light source and a condenser at the top of the stage pointing down, and the objectives and turret are below the stage pointing up. Okay, these are very useful microscope that's generally found in all cell culture lab near to the biosafety cabinet or the cell culture hoods. This slide is basically here to actually show you the comparison between an upright microscope that probably you might have been exposed to and comparing it to the inverted microscope. So as you can see, upright microscopes have their objectives placed above the stage where you put your sample, while inverted microscopes have objectives below the stage where you put your sample. So why exactly is this important? Why is it necessary? Okay, so inverted microscopes, as you know, are actually very much used in many applications in life sciences. Okay, so one example is cell culture. All right, why? Because cells are often grown on the bottom of the dishes, and then they are filled with an aqueous solution. So they cannot be really seen clearly from the above, right? So inverted microscope designs provide several advantages for cell culture applications particularly. They allow easy access to the culture vessels. The objective lens can be positioned very close to the cells, enabling high resolution imaging. And it also minimizes the need for long working distance objectives. So it's really useful, a very useful equipment. Okay, and because inverted microscope in the cell culture applications allow us as scientists or researchers to observe cells in a more natural and very undisturbed environment. The cells can be cultured directly at the bottom of the culture dish or on a cover slip, and it allows for very easy access or manipulation of the cells during experiments. So you can literally have an inverted microscopes in a biosafety cabinet and actually manipulate your cells on the microscope. Okay, so the design is very suited. Okay, so this setup also enable you to use certain specialized cell culture techniques such as time-lapse imaging, right? You can just put your flask there. They can do time-lapse imaging for you, and where your growth and the behavior of the cells that you are growing can be monitored over extended period of times. Okay. Furthermore, the inverted microscope provides much better depth of field due to that larger working distance compared to upright microscope. Okay, this is why is this important? Okay, this is important because particularly when you are actually working with three-dimensional cell cultures, or you are observing cells in a very thick culture medium. Okay, so these are key properties of inverted microscopes. It allows that objective lens to be closer to the cells, resulting in improved resolutions and also clarity in the images that you are getting. And also, another thing is that besides your cell culture flask, um, the typical one that you'll be using in your practical session, there are also other specialized cell culture vessels, such as multi-well plates, microfluidic devices. They are more suited to be used in the inverted microscope compared to the traditional upright microscope. Now we move on to the non-clean area or the non-sterile area. There are four sections in the non-sterile area, which include preparation, wash-up, sterilization, and also storage. So in the preparation area, generally you have a workbench. Okay, a workbench is the area where you do all your preparatory work before you actually go to the laminar flow hood or the cell culture hood. Okay, and then you have weighing balance. They are basically used to prepare your media, reagents, you weigh out powders and things like that. And you have a centrifuge. Okay, so what is this centrifuge? Centrifuge is also one key equipment where sometimes your cell suspension, it actually requires centrifugation because you want to increase the concentration of the cells, or you want to wash off some reagents that you have added in your cell culture. So ideally, a centrifuge is always seated on its own table or bench, far away from other equipments, because they do not want that vibration that's coming from the centrifuge to affect all the other potentially sensitive equipment. Okay, there are many kinds of centrifuge, many sizes, many models available. They even have accessories such as your rotors. Rotors, that is they are interchangeable rotors. They have the swing out, the fixed angle, so they have different size buckets. Okay, so all variety types because they want to be compatible, to accommodate the different kind of tubes that you're using or plates and things like that. Okay, so they are generally very useful equipment, always found in all cell culture labs. For our case, we use the adapters that can adapt to the standard 15 mil tubes or the 50 mil tubes. Okay, and they also have the refrigerated versions because sometimes when you centrifuge for a long period of time, heat is being created. Okay, so you do not want to have that heat to kind of affect your cells, so you have the refrigerated versions to control the temperature within the centrifuge. And lastly, you have the hemocytometer. You'll be using a lot of this. It is for cell counting. So it's a counting chamber device that you can actually use to count the number of cells that you are growing in your cell culture flask. Okay, then you have the wash area, okay, or the wash up area. You have water bath usually is nearby there because water bath is, where you heat up your media or any kind of reagents that you need to heat up to 37 degrees Celsius before you actually use it to your cells. And it's wet. Generally, it's very wet. So generally, it's always near the sink area. Okay, the sink area is where you wash your hands or you rinse your lab consumables. Generally, it comes with the soap, sanitizer, and all your washing gadgets are found there. The sterilization area is generally in another room. It's not in the same room where you do your cell cultures. Okay, what is found in the sterilization area? The main key equipment is the autoclave machine. Okay, an autoclave is a self-locking device that you use to sterilize all your instruments or any kind of non temperature sensitive liquids that you need to sterilize before you use in your cell culture. Okay, and they sterilize it by means of steam pressure. Okay, so sometimes an autoclave machine or autoclaving, and that word itself is also called steam sterilization, where they actually use pressurized steam to kill all the different kinds of infectious agents and denature any types of unwanted proteins. All right, so you use this to reduce the risk of contaminations by sterilizing any kind of apparatus that you may need at very high temperature and pressure. Okay, so it basically kills all the kind of bacterial endospores and things like that. So pretty much wiped out all your contaminants. The sterilization process usually takes about 15 minutes, and they sterilize all your reagents and equipments at 121 degrees Celsius. You can find two types of autoclave machines. You can have the huge one where it is a large capacity floor standing autoclave, or you can find those small horizontal laboratory autoclave that you can put on a bench. Okay. For the storage area, you need lab cabinets, you need fridge, freezer, and also liquid nitrogen storage vessel to store your cells. Okay, so for storage of media and additives, at least you need to have one refrigerator, minimum. Okay, ideally, all the unopened media should be kept separated from that is you're currently using. Okay, they are generally put in a cold room or a refrigerator nearby. Once open, each of these bottle of media should be labeled with the username and also the date that you open it, as well as the cell line that you're going to be using it with, to ensure that both the medium is used within 1 month and also that it is not mixed between the users and the cell lines to prevent any kind of cross-contamination of cultures. Generally, the refrigerators should not be glass fronted because there are many media, they are very light sensitive and they may be degraded by exposure to UV. Freezers at both -20 and -80 degrees is essential because you use it to store media components that are unstable at higher temperatures. You usually create aliquots in small volumes and they are stored separately from the unopened stocks. Okay, but of course you have to clearly label the contents, the date, and also the lot numbers. Self-frozen under suitable conditions can be stored in short term for a few days, but generally if you store it between like fridge 4 degrees fridge to -80, they will definitely lose their viability. Okay, so generally we don't usually freeze cells in like a -20 -80 kind of freezer. What we do is we put them in extremely low temperature freezers or a liquid carbon dioxide sorry a liquid nitrogen storage vessel to maintain these cells for a very long time. Okay, so these cells stocks are usually kept in small little vials and they are kept in such a system or a storage system. This is my last slide, and over here I'm sharing with you all the different kinds of equipments that you can find in a cell culture lab, and it has been divided into the basic requirements, meaning you have to have these items in a cell culture lab, the non-essential ones but beneficial. You have to have these, but if you don't have it, you can kind of borrow from another lab, but you do need some of these items to actually help you during your cell culture work. And also the last one would be the set of equipments. There are useful additions to the lab. So if you have a big lab, you can have space for these types of equipments. You can have them. So they are just useful additions. So what I would need you to do is to take time to do your own research, to pick some of the equipments that are listed here that I have not mentioned, to for you to find out what these equipments are used for. Okay. So with this, I conclude the section 1.2 of topic 1 of cell technology. Hello students, this lecture will cover the last point, which is 1.3 on basic maintenance of cell culture lab. The objective of 1.3, which is the basic maintenance of a cell culture lab, is to discuss the care and maintenance of the various equipments in the cell culture lab. Like all machinery, lab equipment requires regular maintenance to ensure that they're able to function properly and safely. Practicing proper equipment maintenance procedures, scheduling, and record keeping is really essential in order for you to protect your research and equipment investments. Taking care of your instruments actually allow us to really make sure that our instruments will continue to function properly, and it also increases the span of the equipment's useful life cycle. So you do not want to wait until serious problem arises and then you try to fix it. Therefore, you actually do a proper maintenance to ensure that you are promoting the productivity of your work, you help to reduce contamination, and also you help to improve safety. There are three key equipments in the cell culture lab that require good maintenance and also attention, particularly by the lab managers and researchers. The first one is the cell culture hood. Airflow actually is being checked every 6 months because it allows this monitoring of airflow to ensure that you have proper ventilation, which is by now that is critical for maintaining the air quality and also preventing the buildup of any potential contaminants. So regular checks really help to catch any issues early on. Okay. Secondly, you do some kind of a fumigation because it is done to eliminate any kind of pests or pathogens that may be present in the environment. and it is an important step for you to maintain the hygiene and preventing the spread of any kind of diseases and contaminations. Third one is the change of the HEPA filters. HEPA filters are basically designed to trap very small particles, dust, pollen, microbes, everything in the air. So regularly changing these filters will ensure that they remain effective at capturing all these contaminants and hence maintaining the air quality in the cell culture hood. Of course, this maintenance of cell culture hood is really critical because you know what is the purpose of cell culture hoods, right? They are used to provide us a sterile work environment, particularly when you're handling hazardous materials or you're working with any sensitive samples. So this regular maintenance ensures that the cell culture hoods function properly and provides that necessary protection for the laboratory personnel, the environment, and also the cultures that you're working with. Second is the incubators. The incubator chamber is designed in such a way to minimize the risk of contamination. So the design is really pretty unique. Ideally, there will always be this smooth inner casing and the shelves can be easily removed to allow cleaning and disinfection. Many manufacturers offer incubators that have some kind of like a copper interior and shelving, or they even have the modern ones and automatic decontamination routines, which you can actually use in order to reduce the risk of contamination in the incubator itself. Carbon dioxide concentration should be monitored, and the carbon dioxide levels in the tanks should be enough to sustain the weekends, especially when no one is around. It is critical to maintain an uninterrupted gas supply, so it is advisable for you to have two cylinders connected to the carbon dioxide supply system via an automatic changeover unit, because this will allow you to switch to the second cylinder when the first cylinder, the pressure in the first cylinder drops below a certain level. For humidity, the atmosphere within the incubator is normally humidified by means of a tray of water, which is placed at the bottom of the incubator. So you can actually change and clean this tray of water, but always remember that you have to use a deionized or RO water. Okay, and with regular and frequent preventive maintenance schedule in place to ensure that the tray is emptied, thoroughly cleaned and refilled, as it can potentially be a source of bacterial and fungal contamination. Therefore, it is important that the tray can actually be easily accessed and cleaned. You can clean it with a low concentration of a disinfectant detergent. Temperature-wise, they usually have a sensor that allows you to detect whether the incubator is at 37 degrees Celsius. So you actually should also check the sensors to ensure that the sensors are also working carefully, I mean accurately. Okay, so you have to have regular calibration. So you do a periodic calibration to the incubator to ensure that the temperature settings are accurate and it's consistent. Okay, so sometimes it might involve a certain type of calibrated thermometer that is able to verify whether the displayed temperature showing outside matches the actual temperature inside the incubator. Okay. At the same time, you can do general cleaning and decontamination of the incubator to remove any kind of dust, debris, or spills that could be happening throughout the time when people are actually using the incubator. Okay, so you can do this kind of cleaning and decontamination according to usually standard protocols that are given by the manufacturer for the incubator. Lastly is the autoclave equipment. Generally, there is an inspection by MOM every 2 years. The routine inspection allows this autoclave to be inspected for any signs of wear, damage, or corrosion. Okay, they will check the door seals, the gaskets, the valves for any wear and tear, and replace them as needed to maintain that proper seal. Okay, and there's also cleaning involved where they'll be cleaning the autoclave chamber, trays and racks after each use that is done, because this is needed to remove any potential residue or debris that could interfere with the sterilization process. So there is appropriate cleaning agents that you can use by following the manufacturer guidelines to prevent damage to the whole equipment. Third point about autoclave is the water quality. You have to ensure that the water that is being used in the autoclave meet the manufacturer's specification for quality. Because if you use poor water to do this sterilization, it can lead to mineral buildup and corrosion within the autoclave, which then will affect its performance and lifespan. Okay, and next is the water drainage. Okay, how this autoclave can drain out the used water. So you have to inspect and clean the autoclave's drainage to prevent any clogs and also to ensure there is a proper water drainage during and after the sterilization cycles. You also have preventive maintenance, which are highly encouraged by the manufacturer based on certain kind of scheduling. And these include tasks such as lubricating the moving parts, inspecting the electrical connections, and also checking some of the safety issues. You also have to do calibration because you have temperature to play around with and the pressure gauge to ensure that there is accurate readings during that sterilization cycle. This will actually help to maintain the effectiveness of the autoclave and ensure that you really truly sterilize all the equipments that you are using. Okay, all the sterilization conditions are met, okay, based on the required standards. So basically, these are the key things for the three key equipments. However, you do have to do the general routine cleaning and maintenance for most of the other equipments in the lab, such as centrifuge and water bath. Okay? This slide shows you the overall schematic of what has been covered in all the three lectures. So we talk about the design principles, where you have to have physical separation, minimal traffic, how the hood and incubator and microscope has to be placed together. And then we also talk about the basic equipment and lab requirements, the 7 main functions, the clean area covering sterile handling, incubation, and observation, while the non-clean area covers the preparation, wash-up, sterilization, and storage. And at the end of it all, we wrap up with the maintenance, how and why you have to maintain certain key equipments in the cell culture lab. Here are some questions that you can use to test yourself on some of the content that has been presented in the lecture recordings. With this, I conclude topic 1 of cell technology. Happy studying!